So guys, our sequences are assembled and now we got our genomes, chromosomes, and now we want to identify genes in it. So whenever we were interested in finding genes, we need to talk about probabilities. So let's go ahead and have a background on probabilities in this section. So what is the probability? Probability is a chance of an event to occur, and probabilities, this field derives its origin from the games of chance. For example, playing cards or throwing dice. Like you might have played Ludo, Ludo जिसको हम कहते हैं अपनी language में, and you have seen that वो जो छिक्का होता है उसका. So that thing is a die where it has 6 outcomes, 1, 2, 6, and there is a probability that you can get any number out of it. So that is 1 out of 6 probability for each number. If that die is fair, if it is not loaded. If we toss a fair coin, there are two possibilities. Either you will get a head or a tail. So in any case, the probability is half for head and half for tail. Let's see some formal definitions. So we define outcome, it's an each possible distinct result, whereas an event is a collection of outcomes. So probability of an event E is given by NE by N, where NE is the number of favorable outcomes among the total outcomes N. For example, if we want to identify, or if we want to get a probability of an ace, A in your playing cards, जिसको हम यक्का बोलते हैं अपनी language में. So there are 52 total cards and there are 4 aces in it. So probability of an ace is 4 by 52, which is equal to, if we simplify, it is 1 by 13. In probabilities, we use these letters as P's and Q's. So P is actually the probability of an event occurring. It's the probability of success. Now here is an important concept. So what is the success? So success in case of these probability theory and in case of these probability statistics, success is defined as anything in which you are interested in. For example, if you are interested in getting heads, so obviously probability of those getting heads will be represented by P. So in that case, heads are your successes. Whereas if you are not getting those successes, you are failing. So in that case, this probability is Q, which is the probability of tails in this case, or probability of failures. Sometimes you are interested in tails, then success is seeing those tails if you bet on tails. So it is important to understand that success is how you define the success. We also know that P plus Q is equal to 1. Since probabilities are, they range from 0 to 1. So probability of success, if it is 50%, obviously probability of failure is also 50%. We add them together, it becomes 1. So if you know P, you can get Q. If you know Q, you can get P from it through these equations. If you flip around, P is equal to 1 minus Q and Q is equal to 1 minus P. Probabilities are bound between 0 and 1. So that is to say that an event is either impossible, where probability is 0, or a sure thing, that is P is equal to 1, or something in between. So it can be any value between 0 and 1. We use probabilistic models when we do these gene predictions. So let's see what those probabilistic models are. So before that, we define a model. What is a model? So model is a system that simulates the object under consideration. So you can make a model of anything. For example, if you are doing gene prediction, so before that you will come up with a model which we call it as a gene prediction model. Whereas a probabilistic model is the one in which you simulate the class of objects, assigning each an associated probabilities. So you are modeling something and you are also fitting in the probability of the objects which are present in that model. So in that case, it becomes a probabilistic model. So we'll use probabilistic models in our gene finding situations. How can we how can we model those biological sequences? Biological sequences are strings from finite alphabets of nucleotides or residues. So sequences are coming from either 4 nucleotides are coming from those 20 amino acids. We can model them. We can associate each nucleotide at a particular position with its probability, and in this way we can simulate the whole sequence like this. We can have, for example, the sequence can be represented as x1, x2 up till xn, where x is a particular position, or we can say different state, and then we can have x2, which is the next nucleotide. So within x1 we can have anything like A, T, G, or C. So in the end, if we put their probabilities together, so the individual probabilities, they get multiplied, and then their product gives us the probability of the whole sequence. If we look into this equation, we can see that probabilities of all those nucleotides here multiplied together give us the probability of the whole sequence. If we look into the equation, we have this product sign. So we have I is equal to 1 up till n qxi. So here we are simply multiplying the individual probabilities at each particular position for different nucleotides or amino acids. We are simply multiplying them. Let's elaborate that through this example. So where we have the probability of a sequence ATGC. So total we are having 4 nucleotides in this, and we want to see what is the exact probability of this sequence. So right now we are playing with the fair die. Right now we are considering the normal probabilities which are not biased, which are just fair. So under no biasness, we expect 1 out of 4 will be 1 out of 4 times we will see A, and same way T and G and C. So everything is 1 by 4 here, and we want to see the probability of this sequence that is made up of 4 nucleotides. So we simply get the product of their individual probabilities, which is 1 by 4 or 0.25. So when we multiply it together, we will get this number. So that is, we can see this ATGC with a probability of 0.0039. As many nucleotides are added, more terms are multiplied with one another. So since these numbers are probabilities are small, these are coming from out of 1, so we generally try to take their logs in order to bring them into their whole numbers. Here we are modeling rolling of a die. So there are total 6 parameters or outcomes, P1 through P6. So we have like 6 probabilities associated with it, where P1 is the probability of getting 1, and up to P6 is the probability of getting a 6. So probability of rolling I is Pi. We can write it in this general form, and parameter Pi should satisfy that. This Pi is anything between 0 to 1. So probabilities of all those 6 numbers, as long as they are fair, it will be 1 out of 6 or 1 by 6. And sum of their probabilities, sum of all these 6 numbers, that equals to 1. So you add 1 by 6 times, it will go up till 1. So total probabilities, they sum to 1, and individual probabilities, they range anything between 0 to 1. So probability is the chance of an event to occur, and probabilities, they range from 0 to 1.